morning, good evening to all of you across the world. I welcome you all on behalf of the Deborah Rice movement, or in short, DRM. DRM is all about giving voice to the voiceless and hope to the hopeless. Through our helpline, we counsel and pray for the depressed, oppressed, abused, and those with life-pressing issues. Through the contact form on our website, deborahrice.org, people across the world can contact us for counseling and prayer. We have trained counselors and all our services are free. We have also started scholarship for the poor and abuse victim supports recently. Today, the September 10th is the World Suicide Prevention Day. That is the very reason we have arranged for a talk on evolution. You might wonder why. Viktor Frankl, who survived concentration camp and wrote the best-selling book, Man's Search for Meaning, brought out the idea of logotherapy that says that the most powerful motivation for humans is the drive to find meaning in life. Some of his famous quotes are, "Those who have a why to live can bear with almost any how." Life is never made unbearable by circumstances, but only by lack of meaning and purpose. Many people today in life often feel a sense of meaninglessness and emptiness all around them, and try to live for a while without a purpose, and suicidal thoughts may arise at that time. So on this Suicide Prevention Day, I want to stress the fact that there is a purpose to your life. Even behind your pain, there is a purpose. God has made everything with a purpose and plan. The self-proclaimed atheist and one of the biggest supporters of Darwin, Richard Dawkins, believes that the universe that we observe has precisely the properties we should expect if there is at the bottom no design, no purpose, no evil, no good, nothing but pitiless indifference. DNA neither cares nor knows. DNA just is, and we dance to its music. He says in one of his interviews that Darwin removed the main argument for God's existence, since the undirected process presented by Darwin can explain the appearance or illusion of design. Hence, there can be no designer. And if there is no designer, there is no God. And belief in God is some sort of subjective superstition. He calls it the God delusion. Darwin has ruled out the evidence for God. This statement has influenced many of our youths. Many youths in their pre, post, and college years are losing their faith as they go to universities by this sort of message that there is no scientific evidence for God, because this is one of the powerful reasons that many are losing out their faith. While writing my book, Beyond the Boundaries of Science, Exploring the Cosmic Story, I did a lot of research to find out if evolution is scientifically true, since I had a great debate with my son who was doing his medical studies at that time. I found a lot of flaws in the theory of evolution. However, my PhD is not in the field of biology, but in aerospace engineering. Recently, I came to understand that Jonathan, who has done his PhD in evolutionary biology from the Biosciences Institute at Newcastle University in the UK, is speaking about the scientific challenges to evolutionary theory. He's a writer, international speaker, debater, assistant professor at Sattler College, Boston, and a fellow of the Discovery Institute. Hence, I requested him if he can speak in our forum, and he gladly agreed to speak. Before making him to speak, let me give you a brief introduction to this topic. Everyone agrees there is a lot of beautiful and intricate design in biology, right? Why do houses run very fast? Because of their huge lungs and heart that function something like that of a turbocharger in your car with the capacity to extract a large amount of oxygen for its energy. Camels have an amazing feature, and not one, many amazing features that are designed to help them cope with harsh desert environments like non-sinking feet, 3 eyelids and eyelashes to protect them from sandstorms, and so on. Elephants have a unique trunk that can lift loads up to 350 kgs. Giraffes have a unique neck that can reach to around 2.5 meters in length. Dogs are designed for smelling and to run fast. Penguins are underwater flyers. They could survive in seawater due to a gland called supraorbital gland that removes excess salt from their blood. Birds have specially designed wings for aerodynamic lift. History says that the Wright brothers copied the shape of the bird's wing. Bio-inspired design is a cutting-edge field. The world around us is so beautiful. It's detailed, ordered, and complex that it is unbelievable that such a design can come on its own unless there is a purpose behind it. Life around us seems to be designed by a super intelligent being for a purpose. However, Darwinism says, though everything seems to be designed, it's not really designed because there is an unguided, undirected process known as natural selection acting on random variations or random mutations in DNA that can produce the appearance of design. In other words, the unguided natural mechanism has the creative power to mimic a designing intelligence. That's why you can have a design without a designer. The idea of a design without a designer goes back to Darwin himself. In fact, in his 3rd chapter of The Origin of Species, he lays out the logic of natural selection as a substitute for an actual designing intelligence. Charles Darwin was born in 1809. His father was a successful doctor who groomed his son to follow in his footsteps. Darwin entered one of the Britain's top medical schools at the University of Edinburgh in Scotland in 1825 to study medicine. But he soon found that he was unable to even watch an operation being performed. So he left it, and in 1828, he entered Christ College, Cambridge, England, to become a minister. He soon gave up that idea, and while studying at Cambridge, a tutor told him to take up biology research trip around the world on the HMS Beagle, a 5-year trip that would prompt Darwin to write his famous The Origin of Species that became the backbone for Darwinian evolution. Darwin was not the first naturalist to propose that species changed over time into new species. Lamarck was born much before him, and almost around 60 years before Darwin, he proposed a theory of inheritance. However, both the theories are totally different. While Lamarck thought that organisms changed out of need, Darwin thought organisms changed by chance. Though Darwin published his Origin of Species in 1859, the mechanism of natural selection was not accepted for decades since Darwin could give no evidence. Later, when the field of genetics evolved into its modern form from the initial work by Gregor Mendel, who's called the father of modern genetics, Neo-Darwinism, or the synthetic theory of evolution, or the modern synthesis came into existence. We can say that Neo-Darwinism is an integration of Charles Darwin's theory of evolution by natural selection with Gregor Mendel's theory of genetics. So let's now welcome Jonathan, assistant professor at Sattler College, Boston, and a fellow of the Discovery Institute here to this forum. Hi, Jonathan. Would you tell us more about the modern synthesis or the Neo-Darwinism and help us to improve our understanding of evolutionary biology?
Absolutely. Well, thanks so much for having me here. It's a real honor. I will share my screen here. Hopefully you guys can see that. Okay, so it's great to be here. I look forward to discussing with you some of the scientific challenges to evolution. As Dr. Latha said, I want to begin by talking about the background to evolutionary theory, give a little bit of an introduction to evolution and what the theory is and its historical underpinnings. So as you just heard, evolution by natural selection was first proposed by Charles Darwin in his book, The Origin of Species in 1859. He made a very famous voyage, as you probably all know, to the Galapagos Islands. You can see a map here that shows the location of the Galapagos Islands where Darwin sailed to on the HMS Beagle and made various observations, and most notably his observations regarding the size and shape of finch beaks. So the Galapagos Islands is a volcanic archipelago, which is west of Ecuador in the Pacific Ocean. And he made a number of observations, the most famous of which is probably his observations concerning the varying size and shape of beaks of finches. And you can see in the diagram here the beaks of different species of finches that are reproduced from Darwin's The Voyage of the Beagle. And there were different sorts of finches that had different sorts of different diets. So for example, the ground finches typically feed on seeds. There's also tree finches, which typically feed on fruits and arthropod and trees. There's the vegetarian finch, which typically feeds on leaves and fruit. And then there's the warbler finches, which typically feed on arthropods. And Darwin noticed that some of them had long pointed beaks that made some of them more fit for picking seeds out of cactus fruits. Some of them had shorter, stouter beaks, which served best for eating seeds found on the ground. Warbler finches, for example, catch insects in their beaks that are sharper and more slender than those of cactus eaters. So what is thought to have happened is that finch species that are found on the mainland migrated to one or more of these islands where they encountered different environments. And birds from one island could eventually come to colonize another island nearby. And there was, of course, genetic diversity among those birds. So not all of the birds were exactly the same, and their specific features could be passed on to their offspring. And the diversity included the shapes and structures of their beaks. And some of the diversity was present in the population that colonized each island, and more changes arose once the population became established on the island, so that the birds on different islands became dissimilar from one another and yet shared common ancestors. And so as Darwin proposed, those individuals whose beaks were well suited to obtaining food on that particular island were more fit, or so they were better fed, and so they reproduced at a higher rate than the birds whose beaks were less suitable for obtaining food, because they were more likely to starve and not be well fed. So the number of birds with a particular beak shape increased relative to those with other shapes on particular islands. And eventually the shape of the beak became fixed. That is, it goes from being the lone individual out there with that particular beak shape to becoming the norm that gets established in the population. So all the birds in that habitat had beaks of the same shape, because those were the birds that tended to survive better in that particular environment, because they had easier access to food than those that had different shaped beaks. Meanwhile, the genetically diverse birds on one island were also migrating to nearby islands where similar events occurred. But since the environments on the islands were different, a beak that might be well suited for feeding on one island might not be so well suited to another island. And likewise, a beak shape that might be poorly suited on one island might be more favorable on another. So finches from the mainland in South America generally have the more pointed beaks, and so that's considered the ancestral characteristic, whereas the blunt beaks are the derived characteristic that favored survival on particular islands. So that's a basic introduction to Darwin's observations on the Galapagos Islands, which propelled him to propose his theory of evolution by natural selection. He proposed that this process of the more fit organisms surviving in their environment could be invoked to explain much of the complexity, much of the complex adaptations that we observe in the history of life. As Dr. Latha also mentioned, it's the idea of common descent. All species are related by common ancestors, eventually going back to a single universal progenitor of life. That idea was not new with Darwin, right? That idea had been proposed previously by Lamarck, and also it goes. You can find that idea traceable right back to the ancient Greeks. So it's a very old idea. What was new with Darwin was the idea that natural selection, coupled with chance variations, he didn't know what mutations were at the time, could serve as could explain the appearance of design without recourse to an actual designer. So the beaks of Darwin's finches are an example of what's known as adaptive radiation. And adaptive radiation can be defined as the divergence of a single group or species into a series of distinct but related groups or species, each of them suited to survive in its specific niche. So it occurs frequently in archipelagos like the Galapagos Islands, which are strings of islands that are separated from one another and providing different types of habitat. Some of them newly emerged and lacking predators and competitor species. And adaptive radiation can result in very rapid morphological divergence and speciation, and that's what caught Darwin's attention. Now, the birds from one island were relatively isolated from those on another island, and the number of birds of that species on each island was relatively small, around 10,000. And in addition, the trait was fundamentally important for survival and reproduction. So this is why divergence and fixation often occurs most rapidly on islands. So divergence is where you have divergence from the ancestral population or evolutionary change over time. And fixation is where a trait becomes established in the population as the norm. And that happens by virtue of natural selection, which results in the gene variants that help the organism survive better becoming more frequent over time in the population as the weaker organisms that possess less favorable traits get weeded out and get eaten by predators or starved or what have you. So in the 1970s, biologists studied a population of medium ground finches on one of the islands in great detail. And when a severe drought left only large, hard to crack seeds, 85% of the birds perished. The survivors had beaks that were about 5% larger than the average beak size in the original population. But after the drought, birds with smaller beaks flourished again, and the average beak size of the population returned to normal. So this is what's known as cyclical evolution, where with the dry season, when there was a drought, the birds' beaks, the mean bird beak shape changed over time, and the natural selection favored the larger beak size, whereas when the drought was over, then the beaks returned back to the mean. So this is cyclical evolution or phenotypic plasticity. Okay, so we now know the genetic basis, in fact, for the change in morphology of the beaks size and shape of Darwin's finches. So there's a gene that's known as ALX1 that encodes what's known as a transcription factor, which is a protein involved in regulating expression. And this particular transcription factor affects craniofacial development and is now known to be strongly associated with beak shape diversity. Fixation of the beak shape results in fixation of the genome sequence. So remember, fixation is just where you go from being the first individual that has that particular change to taking over the population, as it were. Your descendants take over the population, and that particular trait becomes the norm that's established. And that obviously also results in the fixation of the gene variant that codes for that particular trait. The ancestral population has variation, and the ones which help the organism to survive better end up becoming fixed. So environments in different locations confer an advantage on beaks of different shapes. So the beaks in each group or species become fixed for that shape. and then the underlying DNA sequence also gets fixed. So, so you can see here, this is the ancestral population has variation. The environment favors blunter beaks, and so the blunter beaks end up becoming more frequent. In this other island, the environment favors pointed beaks, and so the pointed beak shape becomes more frequent over time, and eventually all or most of the birds have that particular beak shape. Okay, so what is evolution? What is the modern theory of evolution? So this is a very famous diagram from Charles Darwin that represents the branching pattern of descent with modification from common ancestors, eventually going back to one or a few universal common ancestors of all life. So the theory of evolution really consists of 6 main components or aspects. We have, first of all, the change in allele frequencies in a population over time. An allele is just a fancy word for a gene variant. And so a variation in the gene that confers some sort of diversity that is known as an allele. And over time, the frequencies of those gene variants in the population vary over time. And there's a number of explanations for that. The most prolific, of course, being natural selection. There's also sexual selection, that individuals prefer certain sexual partners over others, certain characteristics, and so that these traits end up becoming more frequent over time. There's also a process known as genetic drift, which is when you don't have the population sizes necessary for natural selection to be effective, and the population literally drifts. It's a random process, and by chance, you might have a gene variant that wasn't necessarily advantageous, but it just by chance ends up becoming fixed because the population size is small enough for that to happen by chance. Speciation is another key aspect of evolution, which is the idea that so we have species alive today that were not alive in the past. You might have a population of organisms which and a subpopulation within that larger population becomes separated from the larger population. Let's say there's some sort of geographical barrier set up, for example, and over time the species diverge, eventually getting to the point where they could no longer interbreed to produce fertile offspring, and now they're considered to be a new species. Then there's the idea of common ancestry, which is the idea that all of life, whether so you and a banana and the fungus growing in the back of your foot, these are all related by descent with modification, such that if you were to wind the tape of life sufficiently far back, you would come to a universal single progenitor of life that is the grandparent, if you will, the great grandparent of all life that exists today, and that would have been a single-celled bacterial cell. There's the idea of gradualism, which is the idea that life evolves very, very gradually, very, very slowly over hundreds of thousands, millions, hundreds of millions, even billions of years. You don't have a chimp-like animal, for example, giving birth one day to a human. That's not the way evolution works. It's a very gradual process. In fact, according to evolutionary theory, there was never really any first human as such, right? It's all a continuum. In the same way that it's very difficult to pinpoint a specific day in which you became an adult, right? It's a very gradual process. It's a continuum from being a child through to a teenager or adolescent, and then finally an adult. And likewise, it's very difficult to quantify how many hairs it takes on the human face to qualify as a beard, but you recognize a beard when you see one, right? It's a continuum. And likewise with evolution, from a chimp-like ancestor to a human, there was never any first human, but it's a very gradual and continuous process. Then, of course, there's the idea of natural selection, which preserves the successful combinations of gene variants, and it results in those gene variants, that combination of gene variants that confers an advantage to the population ends up becoming more frequent in the population, because those are the organisms that tend to survive better. And the ones that harm the organism or cause it to be more likely eaten by predators or starve to death or freeze to death or what have you, those gene variants tend to become less frequent, because the individuals possessing those tend to die off before they get to reproductive age. There's the idea of random genetic mutations. So random genetic mutations so a genetic mutation is when you copy the DNA. There is a copying machine known as a DNA polymerase, and as the DNA is being copied, occasionally there is an error that is incorporated, and about 75% of mutations are neutral, so they're neither here nor there. They don't harm the organism or help the organism. They're neutral, and then a lot of mutations are also harmful to the organism and actually are a hindrance to its survival. And these ones tend to be purged by what's known as purifying selection, which purges those gene variants that are disadvantageous to the organism. And then a very small minority of mutations are actually beneficial. So you've probably heard of antibiotic resistance and insecticide resistance, which occur from these random genetic mutations, and these are among the rare mutations which are actually beneficial. But although these beneficial mutations are rare, these are the ones that end up becoming selected by natural selection. They end up becoming fixed in the population simply because they confer a survival advantage, and so the organisms possessing those mutations are more likely to survive and then and get to reproductive age, and so the frequency of those mutations ends up increasing over time. I'll stop there. And do you want to move on to the next question, or we can take questions from the floor if people prefer?
And thank you, Jonathan. Thank you so much.
Mm-hmm.
And then now let's come to the next question.
Mm-hmm.
As you said, there are many scientific challenges to Darwinian theory of evolution. One such challenge is that Darwinian evolution lacks the explanatory power when it comes to morphing into major animal forms. No one can say that the natural selection and random mutation process is essentially a very creative process that can produce an unlimited amount of change, and that it is a causal agent for all the forms of life we see today. That's what you were telling. So one of the extraordinary discoveries of the last 30 years in biology is the discovery of nanotechnology, miniature machines inside cells. The cell is the smallest unit of life, and even in one-celled organisms, like a bacteria, we find exquisite machinery like a flagellum. Michael Behe brought out this fact by the term irreducible complexity. This idea applies to any system of interacting parts in which the removal of one part destroys the whole system. So I wanted you to speak more about it. And I found there are many extraordinary little machines in our body. So Jonathan, could you brief out some of those machines? And that can be one of the challenges to Darwinian evolution.
Absolutely. Okay, so here I'm going to show a short animation of the world of the cell, which reveals some of the intricate, exquisite, elegant nanotechnology which governs the show in the world of the cell.
I'm finally just beginning to grasp the complexity of the cell. Are there systems within the cell that go well beyond Darwinian evolution? Some type of cellular technology that drives adaptation, replication, quality control, and repair? What if these new mechanisms have massive design implications? Well, I say, so be it.
Okay, so that is just a little taste of the complexity of the world of the cell. I want to introduce at this point the idea of irreducible complexity, which was actually a test that was originally put forward by Charles Darwin in his Origin of Species in 1859, where he put forward a means by which you could potentially, in principle, falsify his theory of evolution by natural selection. He said if it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Now, in 1996, a biochemist by the name of Michael Behe, who's based at Lehigh University in Pennsylvania, he put out a book called Darwin's Black Box, in which he sought to take up Darwin's challenge. And he put forward this idea that he called irreducible complexity, which he defines it as follows. He says, by irreducible complex, I mean a single system composed of several well-matched interacting parts that contribute to the basic function, wherein the removal of any one of the parts causes the system to effectively cease functioning. So to take a common example from everyday experience, Behe put forward the mousetrap as an analogy to help us understand better what he meant. And the mousetrap, as you can see, is composed of a number of different components. You have a platform, you have a spring, you have a hold down bar, you have a catch, and you have staples. And if any of those parts is missing, you don't have a trap that works half as well as it used to, or a quarter as well as it used to, but is broken. And so how would you put together such a trap one piece at a time, where each of the steps has to confer some sort of advantage? So it doesn't become a better and better trap at catching mice the more parts you add, but the function is actually only realized when all of the parts are interacting together to accomplish that higher level objective of catching mice. And indeed, the trap might still not work if the holding bar is too short or the spring too lightweight. The parts not only have to be present, but have to be very carefully crafted one to the other in order to work together to accomplish that higher level purpose of catching mice. The way that Behe initially proposed to define irreducible complexity was in terms of structural parts that are necessary to work together. My own formulation of irreducible complexity has more to do with the functional integratedness of the system, because one could propose that you could add a component that at first is only helpful and then render it essential by modification or loss of other components. That's known as the Muller two-step model of explaining irreducible complex systems. To evade that objection, I prefer to define irreducible complexity in terms of functions, that you require several functions to work together in order to accomplish a higher level objective. And those functions could be accomplished by different parts, right? The identity of the parts needed to accomplish that higher level objective is not so important as the functions being performed. So that's how I would propose to define irreducible complexity. Now, it turns out that the cell is chock full of irreducible complex machines. I'm just going to go through just a few of those, of many, many hundreds that we could discuss. This is a short animation now of the DNA replication machinery, which is used to replicate DNA to prepare for cell division. DNA is the genetic code, the genetic set of instructions that specifies the sequence of amino acids that form proteins, and also also is involved in regulatory functions in the cell. This is where the information in every one of your cells is housed. And this is how it is replicated by the DNA replisome, which is the copying machine of the DNA replication system.
DNA replication is carried out by a molecular machine called the replisome that pulls apart the double helix and makes an exact copy of each strand. In this visualization, we're going to examine the mechanism for copying DNA. The enzyme DNA polymerase synthesizes DNA molecules, using the original molecule as a template for the production of a complementary strand. The genetic code in the template strand is paired to free-floating bases, matching cytosine to guanine and adenine to thymine. DNA polymerase creates an exact copy of your DNA code, making less than one mistake in a billion bases.
Pretty cool, huh? Now, if you were to knock out any of these components, you wouldn't have a system that works half as well as it used to, or replicate DNA a quarter as well as it used to, but it would be broken. It would be non-functional. So, for example, without the DNA polymerase, which is a copying machine that replicates the DNA molecule, no replication could take place at all. There are RNA primers because the DNA polymerase isn't able to start replicating on its own. So the RNA primers have to be synthesized by enzymes known as an enzyme known as primase in order for the DNA polymerase to be able to start. So these would have been laid down, but there would be no replication beyond that if the DNA polymerase were missing. DNA ligase links the DNA fragments that are produced on the lagging strand. So the lagging strand, as you might have seen in the animation, actually one of the strands is copied the right direction. One of them has to be copied backwards, right? The reason for that is because of the double helical structure of DNA and the bi-orientation of the two strands of the DNA molecule. So one has to be copied backwards, and that is done by laying down primers, which are copied from by the DNA polymerase, and then to form what are known as Okazaki fragments. And then these Okazaki fragments are then ligated together, or glued together, if you will, by DNA ligase. And if the DNA ligase were missing, then the newly replicated DNA strands would remain as fragments and would not be ligated together. And so, again, you wouldn't have DNA replication. There's also a sliding clamp, which is necessary to clamp the DNA polymerase onto the strand long enough for copying to take place. And if you didn't have the sliding clamp, then the DNA polymerase would frequently fall off the DNA template, and it would be too slow for efficient replication of the DNA. If you didn't have the RNA excision enzyme, so these are what remove the RNA primers that need to be laid down. If you didn't have those, then the RNA fragments would remain covalently attached to the newly replicated DNA fragments. If you didn't have the DNA helicase, which is the enzyme responsible for unwinding and unzipping the DNA molecule to prepare for copying. If you didn't have that, then the DNA polymerase would stall because it cannot separate the strands of the template DNA ahead of it. And so little or no DNA would be synthesized. If you didn't have the primase, which lays down the RNA primers, then the RNA primers cannot begin on either the leading or the lagging strand. And therefore, the DNA replication couldn't begin because the DNA polymerase needs what's known as a 3' hydroxyl group in order to replicate, but it can't create its own. It needs to be synthesized by the RNA primer, which lays down an RNA molecule. And the RNA polymerase that produces that, which is a special type of RNA polymerase known as primase, it doesn't require a starting point in order to begin replication, whereas the DNA polymerase does. So without the primase, then you wouldn't have replication either. You also need the single-stranded binding proteins, which actually holds the strands apart and stop them from reannealing while the copying is taking place. And if you didn't have the single-stranded binding proteins, then the DNA strands would reanneal before they could be copied. So again, you wouldn't have DNA replication that works half as well as it used to, a quarter as well as it used to, but it would be broken. So all of these parts need, all these different subfunctions need to happen together in order for the higher level objective of copying DNA can be accomplished. Another example is the bacterial diversum, which is how bacterial cells divide. This is just a cartoon of some of the proteins that are involved in the bacterial diversum. This is the Fitz-Z ring, which polymerizes along the division septum in the middle of the cell to prepare for copying. And you need special tethering proteins like ZIP-A and fits A to tether the fits Z ring to the inner membrane. And if you didn't have the tethering protein, then the zip A, the fit Z would be useless. This division ring would be useless. And if you didn't have if you had the tethering protein, but you didn't have the fit Z ring, then again, there would be no purpose of having the tethering protein. So again, you've got irreducible complexity. These are just some of the essential proteins of the bacterial diversum. A related example is cell wall growth. So during cell division, which occurs by a process known as binary fission in bacteria, it requires the growth or the elongation of the cell wall, which is comprised of what's known as peptidoglycan, as the bacterial cell elongates. And that process occurs by the severing of the peptidoglycan backbone and the synthesis of new cell wall material. So as the bacterial cell is dividing, the cell wall, so the cell actually elongates to prepare for cell division, and the cell wall has to be broken and rebuilt during that process in order to allow the cell to elongate in preparation for cell division. And there are enzymes that are known as autolysins, which hydrolyze or cut the beta-1,4 glycosidic bonds that link the peptidoglycan precursors together in the cell wall. And the gaps that are created are then filled with additional cell wall material. So you have this breaking and rebuilding process that's happening in concert with each other. And of course, these autolysins can result in programmed cell death, which is not good. So these have to be very carefully regulated and controlled. Now, this is just a cartoon showing the process of transpeptidation, which is the cross-linking process of the peptidoglycan precursors that forms the cell wall. And this is accomplished by enzymes that are known as penicillin binding proteins. And as their name suggests, these are actually the target for penicillin antibiotics. So when you use penicillin as an antibiotic, this is actually the target, because if you disrupt cell wall synthesis, then the cell is going to die because of the osmotic pressure. It's going to burst because because the concentration is going to be higher and result in the cell to burst. Okay, so this presents, though, an evolutionary enigma. So consider the following two observations. So critical to the elongation process is the severing of the peptidoglycan cell wall by the autolysin proteins. Critical to cell viability is the resynthesis of the peptidoglycan cell wall. Because the peptidoglycan, you need to be able to break the peptidoglycan cell wall in order for cell elongation to happen in preparation for division, but the cell will die, it will burst, if you don't have the means to rebuild, to resynthesize the peptidoglycan cell wall. So breaking of the cell wall can serve no adaptive utility until a mechanism has also arisen for the simultaneous integration of those peptidoglycan precursors that build the cell wall. Indeed, without the latter mechanism, the cell is rendered non-viable. And we know that how critical the cross-linking process that rebuilds the cell wall is to prevent cell death, because when penicillin targets the penicillin binding proteins, the cell dies because it's no longer able to rebuild the cell wall, and so it dies because it bursts due to the osmotic pressure. Okay, so that's another example of an irreducible complex system. Another example that was made very famous by Behe is the bacterial flagellar motor, which resembles a typical man-made motor. This is a propeller that propels various bacteria through liquid. And it's got a rotor that rotates within a stationary component, or the stator. You've got bushings and bearings. You have a drive shaft or a rod. You have a hook structure. You have a propeller, and these are very efficient machines, right? So these in some species like Salmonella, Typhimurium, or Escherichia coli, these are spinning at speeds of up to about 17,000 RPM, rotations per minute. In other organisms like Vibrio cholera, for example, these can spin at speeds of up to like 100,000 RPM. And These are plugged into a very elegant signal transduction circuit, so they're getting feedback from the environment, so they know whether they're going towards food like glucose, or if they're running towards poison, and they need to change direction. And these things can change direction very, very quickly. They can turn on a dime and start spinning 100,000 RPM in the other direction very quickly. And the signal transduction circuit is very, very elegant piece of technology. But one characteristic of these systems that really distinguishes them and marks them out from man-made technology is not just their virtual 100% energy conversion efficiency, but also the fact that they are able to self-assemble. They actually carry assembly instructions, so they can build themselves from the inside out. And I'm going to show a short animation that reveals how this happens. So that was the assembly process of the flagellar motor that propels bacterial cells. These are different in structure from say a sperm flagellum, for example, which is more like a whip, which beats to propel sperm through liquid, whereas the bacterial flagellum is more like a propeller, and it rotates. And they're very different in structure. Now this shows the filament capping protein, which is known as FlyD, which is this pentagonal shaped disc, which functions like a molecular chaperone. So it basically folds proteins. So these monomers of flagellin, which form the propeller, these have to be, because they have to pass through a very narrow channel, which is like 2 nanometers in diameter. And the channel is so narrow that these have to pass through, that they have to be unfolded for insertion into the channel and then subsequently refolded at the distal growing end. And that is accomplished by this chaperone molecule, which rotates, called FlyD. And experimental studies have shown that if you knock out FlyD, then you lose these flagellin monomers into the medium. And so the flagellum does not assemble. And there's a lot more that could be discussed on this, but I will leave it at that for just now. So irreducible complexity is evidence of design. Why? Well, explaining the origins of irreducible complex features requires a cause that possesses foresight. Only intelligence is able to visualize a complex endpoint and bring together everything that's necessary to actualize a complex endpoint. So intelligent design, I would argue, is the best explanation for these features of life, where we have a higher level objective being accomplished by multiple subfunctions that are required to work together to bring about that higher level objective. So I will close there, and then we can move on to the next question.
Yeah. Thank you, Jonathan, for your wonderful answer to the question on the molecular measurements inside the cells. I think next, let me come to the final question that is related to the tree of life. You can answer it shortly. Next time, we are going to the episode 2, where you can do it in detail, Darwin portrayed the process of evolution beginning from probably one-celled organisms, which then gradually morphed and changed so that all the forms of life we see today are related by common ancestry to forms of life that lived a long time ago. He depicted the history of life as a great branching tree where the root and trunk of the tree represented the first life, the branches at the top, all the forms of life that we see today, and the branches in the middle, that forms that morphed gradually from that simple form to what we have today. So this gradual transition from one form to the another had come under fire recently by a lot of people. Even Sir Fred Hoyle and Chandra Wickramasinghe, many of them have spoken against this. So can you explain how much time you would need to go from one transition to another or one species to another? Can you just give some few idea on that?
Sure.
And summarize your talk.
Yeah, sure. Let me share here. Okay. So here I want to talk about the waiting time problem, which really provides the mathematical basis for the argument from irreducible complexity. Why is it that complex adaptations such as that we observe in irreducible complex systems are prohibitively difficult to accomplish through natural selection acting on chance mutations? Well, the key issue here is that complex adaptations require coordinated mutations. Now, what's a coordinated mutation? A coordinated mutation occurs when two or more coincident genetic changes that occur that only together produce an adaptive phenotypic effect that allows for selection to operate. So one of the mutations by itself doesn't provide an advantage. It's only when both happen together that you have the complex adaptation. So this gives rise to the waiting time problem. So evolution is supposed to proceed by random mutation and natural and sexual selection. But selection can only work in mutations with a positive or negative adaptive value, and at least some adaptive advantages require two or more coordinated mutations. All mutations have two time constraints that depend on population size and generation time. We have the waiting time for a mutation to occur and the waiting time for the fixation of this mutation. So that's it's got to go from being the lone individual out there with that mutation becoming the norm that's established in the population. Now, the question that we can ask is, does the history of life provide the resources for evolution to accommodate these waiting times? So this gives rise to the waiting time dilemma. So with large populations, the waiting time for a mutation to occur decreases, but the fixation time increases. And the reason for that is because in a large population, you have more individuals for the mutation to arise in by chance. So the waiting time for the mutation to occur is going to decrease because there's more individuals for it to happen in. There's more probabilistic resources, if you will. But the fixation time increases because there's now more individuals for the mutation to sweep through in order for it to take over the population. With a small population size, on the other hand, the waiting time for a mutation to occur increases because you have fewer individuals for the mutation to happen in by chance, so you have lower probabilistic resources. But the fixation time decreases because there's fewer individuals for the mutation to sweep through. Thus, there's no easy way for evolution to work around the waiting time problem. This is a dilemma in evolutionary theory. Now, one could respond to this as a potential counter-argument could be that recombination allows for neutral mutations, which account for about 75% of all mutations, to occur separately in a population and to combine later by sexual recombination. However, there was a paper published in 1998 by Christensen et al., which concluded that, quote, recombination lowers the waiting time until a new genotypic combination first appears, but the effect is small compared to that of the mutation rate and population size. So it makes a difference, but not a very significant difference. We can do the math. So the fossil record actually is very useful for providing the data for the available time frame. The standard and the standard formula of population genetics allows us to do the math. All you need to do the calculations are reasonable estimates for the following three parameters that can be established by comparison with recent organisms. We need to know the mutation rate, the effective population size per generation, that's the individuals that reproduce, and the generation turnover time. Now, the waiting time problem was discovered initially by Michael Behe and David Sinoke in a paper published in 2004. And then Michael Behe, later in his 2007 book, The Edge of Evolution, made the argument that the waiting time for 2 coordinating mutations is prohibitive for the neo-Darwinian mechanism of evolution to work. He used in his book the, oops, I should say 10 to the 15 years. He used in his book the example of malarial resistance to the drug chloroquine, and he concluded that it would require more than 10 to 15 years for that to occur, given the known, oops, okay, yeah, so he used the example of malarial resistance against chloroquine drug, which require 2 mutually dependent mutations. So that's malaria, which is a parasite that infects humans and causes malaria. It's known as Plasmodium falciparum. And to acquire resistance to the chloroquine drug, he looked at the amount of time it would require in the human population, and given the population size of the malarial parasite, and then he applied these data on human evolution to work out, okay, how long would it take to require a 2-step coordinating mutation in a hominid population? Because in order for the malarial parasite to acquire this resistance to chloroquine, it required 2 mutually dependent mutations. And so he extrapolated from how long it would take how long it's known to take from experimental studies in malaria to how long it would likely take in humans, granting that we're dealing with fewer individuals and so forth than we are for the malarial parasite. And he calculated or estimated a waiting time of something like 10 to the 15 years for that to arise in a human population, to get this complex adaptation involving 2 coordinated mutations. In 2008, 2 mathematicians called Rick Durrett and Dina Schmidt in the Journal of Genetics published a mathematical model. The title of their paper is "Waiting for 2 Mutations with Applications to Regulatory Sequence Evolution and the Limits of Darwinian Evolution." And they sought to address or even refute Behe's analysis, which was based on experimental extrapolation. And their mathematical model argued that it wouldn't take as many as 10 to 15 years as Behe had proposed. Rather, it would only take 216 million years for that to arise in the human population. But granting that they're correct, and of course, there's in any mathematical model, there's going to be simplifications and stuff and so forth. But granting that they're correct, and it would only take 216 million years, the human-chimp divergence only occurred around 6 million years ago. So it's still it's still prohibitively difficult to get those 2-step coordinating mutations to arise, given the timeframes allowed by the fossil record. And there was also a paper published by John Sanford et al. in 2015. So he used a computer simulation to calculate the following waiting times based on reasonable estimates for an incestuous hominid population of 10,000 individuals and a generation time of 20 years. Because until very recently, you only had about 10 to 20,000 individuals, breeding individuals per generation in the human population. A generation turnover time of 20 years. And so he estimated that the fixation, the time for the specific point mutation to become fixed in the population of humans, would be about 1.5 to 15.9 million years. And for single codependent mutations, it blows out to like 85 million years. So very prohibitively difficult given the time frame of the human-chimp divergence, which is only about 6 million years ago. A more recent paper just published this year by Ola Hossier, Günter Beckley, and Angager also put forward a methodological paper where they looked at methods for calculating the waiting time until coordinating mutations get fixed in regulatory sequences. And the paper opens by observing that, quote, "A classical problem of population genetics is to study the time until new genetic variants first appear through germline mutations and then get fixed, i.e. spread to all individuals of a species as it adapts to a new environment and evolves over time." It also notes that in previous studies, quote, "analyzing evolution of whole DNA sequences of nucleotides of length L written on the 4-letter alphabet A, C, G, and T, the waiting time where each is neutral, i.e. gives no selective advantage, increases either polynomially or exponentially with L." So the paper develops a mathematical model and it concludes that a number of interesting results can be derived from our general setup. For instance, that the expected waiting time increases exponentially with M, for a selectively neutral model where when backup mutations are possible. So the more complex the adaptation, the more coordinated mutations are necessary, the more difficult it is for evolution to accomplish by chance. But it gets worse than this, because changes in amino acid sequence that give a protein a new function must often be accompanied by corresponding changes in regulatory sequence, which determine how and where the gene is expressed. So for example, myoglobin is a protein that's similar to hemoglobin and is used in muscles to help extract oxygen from the blood for use by the muscle. And myoglobin's function is made possible by its higher affinity for oxygen. Myoglobin if it were to be present in red blood cells, it would not provide a selective advantage. In fact, it would be harmful to the organism because it would bind too tightly to oxygen and not release it to the tissues. So it's not that hemoglobin and myoglobin share a common ancestor. There was a gene duplication at some point in evolutionary history, and one copy retained its original function of carrying oxygen around in the blood, and the other give rise to this protein myoglobin that deposits oxygen in the muscles. But if the myoglobin were to be expressed in the bone marrow where the red blood cells are produced, then it would actually be a hindrance because it would bind oxygen too tightly and not releasing it to the tissues. So you need not only to get the amino acid sequence changes to produce a new protein, but also have to get to change the regulatory control. Finally, there's I'll talk about this case study in whale evolution. So there's a number of different changes, anatomical changes, that are required to change a terrestrial mammal that it's thought whales evolved from, and a fully aquatic to a fully aquatic seagoing whale. Some of the anatomical changes are documented in this paper, a phylogenetic blueprint for a modern whale. Some of the changes that are necessary include an intra-abdominal current heat exchange system, because in whales, unlike in most mammals where the testes are outside the body, the testes are actually inside the body next to the muscles that generate heat during swimming. And so what happens in mammals, if the testes fail to descend and they're not cooled, you get sterility, which is of course an evolutionary dead end. So you have to actively cool down the blood, which is done in a manner akin to a modern refrigeration system by shunning cooled blood from the tail flukes to a nexus of arteries and veins that dissipates the heat from the gland. So the testes are kept below core body temperature. You need a reorganization of kidney tissue to facilitate the intake of salt water, because you're no longer just intaking fresh water, you have to intake salt water. You have to have reorientation of the fetus for subaquatic birth, because instead of coming out head first like you would on land, now you're coming out tail first. You have to have modification of the mammary glands for nursing of young underwater. You have to have forelimbs transformed into flippers. You have to have substantial reduction of the hind limbs and the pelvis. The tail has to be transformed into a fluke. You have to have a ball vertebra instead of a sacral vertebra, because the tail, instead of wagging side to side like a dog's tail, has to move up and down. You have to have a special lung surfactant, because when whales dive and come back to the surface, the lung needs to re-expand very quickly. So, and just on, lots of anatomical changes necessary to transform a terrestrial mammal into a fully aquatic whale. According to the most generous estimate in the literature, the time frame that is required the time frame that is granted by the fossil records to get from the fully aquatic terrestrial mammal to go from the terrestrial mammal to the fully aquatic whale is about 9 million years. And you can make an argument that the time frame is actually much narrower than that. So how do you explain this? According to Rick Sternberg, he modified the equations of the Jordan-Smith paper to account for the fact that we're dealing with more individuals and so forth. According to his math, in this population of terrestrial mammals, it would require about 43.4 million years for that to occur. But according to the fossil record, you only have 9. And that 43.4 million years is only for the two-step complex adaptation, where you need 2 coordinating mutations to accomplish a complex adaptation. So the math just doesn't add up as far as the timeframes allowed by the fossil record are concerned. Finally, there's also this case study in bacteria. This is published by Doug Axe in the journal Biocomplexity. This paper called "The Limits of Complex Adaptation: An Analysis Based on a Simple Model of Structured Bacterial Populations." And he says, quote, "Since we now have a large and rapidly growing catalog of functional protein systems that seem to be fundamentally complex, there is a growing sense that innovations of this kind would require complex adaptations, meaning adaptations needing not just one specific new mutation, but several, with all intermediates being non-adaptive. If so, this may present a probabilistic challenge to the standard evolutionary model because it would require fortuitous convergence of multiple rare events in order for a selective benefit to be realized." And this is a graph from his paper, which basically shows that if you require, if the first mutation is neutral, so it's neither, it doesn't cause a harm to the organism, then the most coordinated mutations in a bacterial population, which have massive populations, is 6 or fewer. Whereas if the first mutation is maladaptive, it causes some harm to the organism, then the highest number of mutations that the complex adaptation can require is 2 or fewer. Okay, I'll finish with this one. So this is then the problem of reductive evolution. So the Darwinian model of evolution holds that one of the key mechanisms of evolutionary innovation is the duplication of genes and the subsequent divergence of one of the duplicate copies to undertake a new functional rule. So it's widely believed that a duplicate gene has no phenotypic cost, no cost to the organism, or advantage associated with it. So it's selectively neutral. And in such a state, it's thought that the gene is free to mutate, independent of selection constraints or pressure. And when a previously protein-coding gene incurs deleterious mutations such that it no longer codes for a useful protein, the gene is rendered a pseudogene. So because the probability of a single gene stumbling upon a significantly different yet functionally advantageous sequence is so small, the idea then is that following a duplication of a gene, one copy is able to retain the original function, while the other is free to explore combinatorial space in search of some novel function. Gauger et al. 2010, this is a paper that was published looking at populations of E. coli, and they broke a particular gene in E. coli called TRPA, which codes for an enzyme involved in the biosynthetic pathway leading to the production of the amino acid tryptophan. And the E. coli are no longer producing that amino acid tryptophan. And they broke it in 2 places so that there's this two-step pathway, simple two-step pathway leading from no function to partial function to full function. So one of the steps, one of the mutations would have resulted in partial function of producing tryptophan. The other would have resulted in full restoration of that pathway. Now, in the study, the TRPA gene was broken so that it could recover the ability to synthesize the amino acid tryptophan by reverting only 2 single-point mutations. And what the authors found is that none of the lines of E. coli actually took the pathway. In fact, they broke the gene further. Because the energy cost of transcribing a broken gene is high, so they broke the gene in order to confer the advantage of not having to transcribe a broken gene. None of the lines of E. coli took that 2-step adaptive pathway to tryptophan biosynthesis. So the authors conclude that the cost of transcribing a broken gene incurs significant fitness costs, and thus its removal is actually facilitated by positive selection. Another study published the same year, 2010, I think it was, McLatchie and Nachman, titled "The Extinction Dynamics of Bacterial Pseudogenes," concluded a very similar thing. They examined the genomes of Salmonella species, and they argue that the evolution of bacterial pseudogenes is not an entirely neutral process. Rather, bacterial pseudogenes are often actively degraded by the forces of selection. And degradation of genes which don't code for proteins or perform regulatory functions is most probably due to the energetic cost of transcribing them into messenger RNA. So we can see then, in summary, that evolution has a lot of problems from multiple disciplines within the life sciences. I think that the evidence actually points more towards a design than to the neo-Darwinian scenario of natural selection and chance mutation. So I'll conclude with that. Thanks for your attention.
Thank you so much, Jonathan. Thank you all for listening to it. We took up a few scientific challenges to neo-Darwinism, one such as molecular machinery in our body, And the next one is a time problem for change from one species to another. And Jonathan beautifully explained. Now we will take a few more challenges in our next second and the third episode. Though there are many scientific challenges to Darwinian evolution, today I see many Christians accepting that as good science. Their presupposition is that evolutionary theory in no way conflicts with, nor does it deny the existence of a divine creator. Some advocates go even further. They see Darwin as a friend to faith. So regarding this, I have a philosophical question to one of our panel members. We have Prem Isaac with us as one of the panelists. Prem worked in software development and data science for the past 29 years with undergraduate studies in physics and computer science. He also has a master's in philosophy and has served as the international director for Latha Christie. He has taught apologetics. Let me pose a philosophical question to him before moving on to addressing the participants' question. Prem is positing God as the design agent, or God's creative powers at the junctures in life history that science cannot so far explain, amount to the fallacy known as putting God in the gaps, or the God of the gap fallacy. Can science find mechanistic explanations for every presumed miracle? Can you give a short answer to it?
Okay, great. Well, thank you for having me on this discussion forum on your panel. I think it's a great question. It has been often alleged that invoking God as a cause for some event in the natural history of the world, whether it's the history of life, as we are discussing in this case, or perhaps even the history of the universe, is somehow a fallacy, a wrong way of thinking, known as the God of the gaps fallacy. So for us to understand when it is a fallacy and when it is not a fallacy, we need to make a distinction between the kind of science that we are trying to do through something like the theory of evolution, and the kind of science we're trying to do when, for example, we build a car or when we build an airplane. So the ways of dividing this would be to distinguish operational science, which describes regular repeatable processes in nature. For example, the evaporation of water or rainfall, or even earthquakes or lightning, for example, which happen all the time and are frequent. So on the one hand, these can be studied by observation. That is to say, the events in question can be seen or detected by the person doing the investigation. And these events are repeatable. They many times lend themselves to experimentation, where we can even generate the event and do it repeatedly and be able to study it. So we call this operational science. And then we have origin science, which describes non-repeatable processes, which are abrupt and discontinuous and cannot be observed directly. So as an example, the origin of the universe as a whole, or the virgin birth in Christianity, the incarnation of God through Christ, or for example, the resurrection, or anything that we would refer to as a miracle, which typically is abrupt and discontinuous and really cannot be replicated. So if we call something to be a miracle, but we are able to repeat it over and over again, then somehow it is not, doesn't look like it's actually a miracle. It's just something that's subject to the laws of nature. However, if you think about something that is not repeatable, an example would be perhaps a murder investigation, wherein the person who's trying to solve, a detective, for example, who's trying to solve a murder, cannot go back in time to observe the event of the person being murdered. they cannot repeat that action several times to be able to observe it. They can't even observe it once. What they can do is look at the evidence left behind and attempt to create an explanation that will fit the evidence. So that would be called origin science. So evolution is an attempt at this type of reconstruction of an explanation of events that have taken place in the past, which are not directly observable. Now, we are not referring here to the level of change that we can see as an example through natural selection when we're thinking about the changes in the shapes and sizes and functionality of the beaks of the finches, for example, because that is obviously something that we can observe. But we're talking about what we would call macroevolution, where the idea that Darwinian process can account for really the entire range of biodiversity within the time period that evolutionary theorists believe these changes to have happened. So that's not something we can directly observe. So the way to think about when is it okay to invoke God and when is it not okay to invoke God, or even if we didn't consider God, at least an intelligent agent, is that when we're thinking about operational science, that is to say, regular, repeatable events, things that we can observe, and we end up invoking that God must have done something just because we don't understand it. We will end up giving misleading ourselves and our hearers, and also giving the idea of religion a sort of a bad reputation, because one day some natural explanation will be found, and then those who have invoked God will look silly or foolish or something like that. For example, if somebody says, well, because we don't know why it rains, there must be a rain god, and because we prayed, the rain god sent rain, or we offered a certain sacrifice or something like that, and that's what causes the rain to come, is that there's some divine being that's sending the rain. Then we find out later on that rain is nothing but water that evaporated from the ocean and the rivers, that goes up into the air and cools down and forms clouds. And then when the clouds become concentrated and thick, they eventually disintegrate, and the rain in the cloud, the water in the clouds just comes down. So now that a naturalistic explanation has been found, those people who have invoked God as the reason appear irrational or emotional or silly, or even worse, stupid or even deceptive. So in a situation like that, where the phenomena are repeatable and they happen with some regularity and they're easy to observe, we should refrain from just jumping to the conclusion that it has to be a divine agent that would have had to be responsible for that occurrence or that event. However, when we're talking about origin science, wherein something happens abruptly and it's not repeatable, we have to look for the best possible explanation. So there, the laws of nature, we end up not being able to explain something through the known laws of nature. We run out of resources. And a simple example would be the origin of first life. Even the theory of evolution does not even attempt to handle the question of first life. That has nothing to do with Darwinism, actually. Darwinism assumes that there is life, and then aims to give an explanation for how diversity occurs within the life forms, the different life forms that we see. How do we account for the fact that there are different kinds of life forms? But it doesn't really handle the question of where did life come from in the first place. So this is an example of an origin event. And if there is especially a need for information. And I think that's the key. That's the way I would look at today's session is many of the things that Jonathan presented have to do with the necessity of information, which may include things like foresight, wherein there must be an intended goal towards which different mechanisms appear to work simultaneously. So this type of existence of information and its usefulness, it's not just sort of static information, but information that gets used in order for the processes of life to work. And many times there's these processes which are simultaneous. It's very difficult to see how natural law can explain such processes. And so here, if we invoke intelligence or God as an intelligent agent, that is not a God of the gaps fallacy. Once you run out of naturalistic resources, the only option left is to invoke intelligence. We do see intelligence and its effects all the time, since we are, as humans, intelligent, and we produce effects which cannot be otherwise produced. And so there is a point of comparison as to saying, hey, these effects are produced by natural causes, but these other effects are produced only by intelligent causes. And so being able to make that distinction is how we can tell that a miracle happened. So if virgin birth was just happening all the time, we would not refer to it as a miracle. We would almost codify it as some sort of law, that law of nature. But because virgin birth doesn't happen all the time, we are forced to think of why any even single instance of virgin birth, why it could happen. And clearly that is a pointer towards the supernatural. So just as God of the gaps, invoking a God every time we don't understand something can be a fallacy, that is to say, a wrong way of thinking. There is another equally wrong way of thinking, and called nature of the gaps, which we don't hear about too often, coming from especially in the academic community. So the nature of the gaps fallacy is nothing but a sort of a blind faith in nature. So whenever there in the theory of evolution, there is some sort of gap, so something that we are unable to explain, instead of being open to the supernatural or to intelligence, more generally speaking, we jump to the conclusion, or we have the temptation to say that, well, one day we will find a natural cause for it, and somehow Mother Nature will give us an explanation. We will somehow find one. So it's sort of a blind faith in Mother Nature, even within the origin science, where we are unable to actually repeat or explain. There are people who exercise this blind faith. So what I'm saying is that is also a fallacy. It's a nature of the gaps fallacy, which is the assumption of naturalism. That is to say, just from the beginning, when we start thinking about these things, even before we look at anything, we've already decided that we cannot permit intelligent agency. We cannot allow a divine foot in the door. We cannot think about even the remote possibility of there being a divine agent. So even before we begin to do this, we sort of decide to have an anti-supernatural bias. And then therefore, anything we cannot explain, we state that, well, one day we will find a naturalistic explanation. So I think that is equally a wrong way of thinking. It's as invoking God for every single thing.
So now, thank you so much, Prem, and thank you, Jonathan, for coming over here and explaining the questions posed to you. So now let's go to the participants' question. There are a lot of questions today, more than 100 joined, and now we have around 60 to 70 with us. Let's go to the questions that are posted in the chat. I think the first one is for me. Someone has asked Grace Vinod Kumar, biblical timeline of a few thousand years. how it will fit in the million years of evolutionary biology. And another person also has asked how the biblical timeline of 6,000 years can fit in all these things. But my answer to that is, how do you know that Bible says it's 6,000 years? Because you take it literally that evening and morning. So in 6 days, God created, and then he comes to the human beings, and then you calculate, and then you think that it is 6,000 years, right? This was brought forth by Usher, one theologian, and he calculated and told everything came just 6,000 years back. But actually, if you read the Bible, why can't it be a revelation? You go to the last chapter of the Bible, it's a revelation. And the first chapter, before the humans were created, how did they come to know that how the creations happened? It should have been a revelation to someone else. That's what I have brought in my book, Beyond the Boundaries of Science. I brought in a new model called the Revelatory Day model. In that, I have just told for example, you can take even Moses. He was there in at one point for 6 days when the clouds covered him. So God could have revealed on the first day how the light happened, how the photons were released, and how the whole universe were lit by the light because of the stars that were burning forth and supernova explosions. And then next, you can see the firmaments forming, and that how the earth was beautifully put in that protective environment. And the third, like one by one, and then the moon forming, and then fourth day you can see. So this can be 4 days of revelation. So then you need not worry about the timeline. God never spoke about the timeline. Even I used to wonder at the prophets who say that the world is going to end. God never wanted us to find out how many days this earth will be there. Science clearly says one day the whole universe, the whole galaxy and our solar system will collapse after many billion years. So we don't know what's going to happen next. So let's not worry about the number of days. Let's come to the understanding today. As I said, the initial part of my talk today is World Suicidal Day. They believe there is no purpose. Many of the suicidal victims, when you ask them, they say there is no purpose in their life. So that's why we have to tell them there is a God, because why there is no purpose? God brought them into existence with a purpose. Even the pain has a purpose. So when there is no God, there is no purpose. So why God is removed? Because everything happens by chance. So that's what Jonathan spoke, that everything did not happen by chance. There is a design behind everything. So that's what I would like to answer that question. Now, next, the question I will give it to Jonathan. And then moving from lower order forms to higher order organisms was not by evolution. Then how that process came into progress, whether God, it's an act of God at every point, or what do you think about it?
Yeah, so in terms of the process of evolution, I would reject the naturalistic scenario. I think that there is evidence of design. I think that the complex adaptations we see in the history of life are better predicted on the design hypothesis than the Darwinian hypothesis. What was the other question?
What's the other question directly exposed to you? Is it wrong to consider evolution as the best naturalistic explanation when you bring in design being necessary to explain irreducible complexity? Does it not shift from science to philosophy? How do you scientifically conclude that design could be the only explanation? I understand that this is a science bias. What do you think about Dr. Joshua Swamida's book and Dr. Cray's book, which tries to reconcile evolution with theology?
So in terms of my evaluation of Dr. Swamidas's book, I think it's a bit of a train wreck, to put it mildly. If you want my reasons for that, you can go on my website. I have an article where I discuss Adam and Eve, and I give a critical appraisal of Joshua Swamidas's approach and argue why I think it's not at all convincing. In terms of, is it wrong to consider evolution as the best naturalistic explanation? Well, you could say it's the best naturalistic explanation because on naturalism, it's really the only game in town. So of course, it's the best naturalistic explanation. In terms of categorizing intelligent design as science or philosophy, I would be inclined to categorize it as science, although obviously with philosophical underpinnings. because it's based on empirical data. Intelligent design, I would argue, makes robust predictions about what we would expect to observe in the cell. We expect to observe irreducible complexity and specified complexity and these sorts of features that we habitually associate with design. I think that the question of how to categorize intelligent design is really a red herring, because the more interesting question is whether it's true, not how do we categorize it. So so hopefully that addresses that question. And I think you asked, there was another question you asked, but I forgot what it was.
Science today seems to indicate that humans have been on the face of this earth for around 2 billion years. So is Adam a selection from among the humans that existed in Mesopotamia, or is Adam figurative?
I don't know where this person's getting the 2 billion. Did they say 2 billion? Because that's not correct. I mean, animals didn't exist that long ago. It was back then, it was only bacteria that lived on the earth. I mean, there's obviously different species of hominids. Humans, in terms of the genus Homo, go back, a good couple million years. In terms of Homo sapiens, they have been on the scene for something like 100,000 or 200,000 years. I would argue that the population genetics data is consistent with all humans being derived from a single primordial pair that lived over 100,000 years ago. So I don't think that the scientific data is consistent with, I don't think the scientific data is inconsistent with us being derived from a single primordial pair. But even if it were, I think that a fallback position that would be quite plausible is to suppose that Adam and Eve were the first priest and priestess of humanity, and that they represented the initial founding human population. I think that that is a legitimate view that one could take if it turned out that there was strong evidence against the plausibility of a primordial pair.
So another question again to you, did a single cell organism just appear in the beginning, and from that other organisms evolved? Or was there an explosion of life at some time? There is fossil evidence of the Cambrian explosion. How did that occur? Actually, we are going to cover Cambrian explosion in our next episode. So, so would you like to answer now, or I think we can do it in the next episode?
Let's hold off that for next time.
Yeah, so that's okay. So kindly join for the next, and we are going to cover step by step. Kindly ask question from what we spoke today. Tell us more about the mitochondrial Eve.
I'm not sure what he wants to know about it, but so the mitochondrial DNA is passed on down the maternal line. And so we can use mitochondrial DNA to trace back ancestry through the female line, because mitochondrial DNA is passed down through the female. There's mitochondrial Eve is the last common ancestor that's universal to all humans, the last female common ancestor that's universal to all humans. And there's also Y chromosomal Adam, because Y chromosomal DNA is passed through the male lineage, and so we can trace it back to a Y chromosomal Adam, which is the last common ancestor of all males, the last male common ancestor of all humans. Hopefully that answers your question.
Yeah, Jonathan, this is the question they have asked directly to you. There is a certain snake called the spider-tailed horned viper. This viper is generally well camouflaged because of its color, but its spider-shaped tail is what helps it to attract small prey that could feed on. During the process of evolution of its tail, is it right to assume that the tail underwent a process like first changing color, then size, and over a long period of time, slowly changing to the shape of a spider? During the change of the tail, doesn't it make the snake exponentially more vulnerable for bigger prey because of its tail? If that is the case, how does the tail then continue to evolve into the current shape of a spider? Shouldn't they die out due to the tail being a liability during its change? On a similar point, what reason is there for evolution to bring in beauty? So I think this is one point for the transitional fossil, because transitional forms we wanted to speak again in the next episode that it is very difficult to survive many of the transitional forms. That's what he's telling. You can answer if you have something on that.
Say that again, that last thing again.
No, they are asking about the transitional forms, like from snake to the spider, why it doesn't survive or something. This is all about transitional. So would you like to speak, or which episode you are covering the transitional forms?
Yeah, so I'm not an expert on this, so I don't want to comment on things I'm not an expert on.
Okay, so kindly post your question based on the talk today, because we would like to cover it in 3 to 4 episodes. And so what about the fossils of early humans? Are they early humans or a different species that is now extinct as they look similar to humans? See, this is also, we are going to speak about human evolution. So I would like to say, what about the fossils of early humans? Many of the fossils you can read in my book, Beyond the Boundaries of Science. I've written about the fossils, how either they are found to be apes or they are found to be some humans. So there were no transitional forms. Most of the things, when they went into detail, it's based on your worldview. You can construct how you are constructing is based on your worldview. So I think he will cover Jonathan in detail in one of his talk. Is my book available online? Yes, you can just type. It's also available in Amazon.com and Amazon.in. So you can just type in my name and you can get the book. So I'm going to unmute everyone. So for the next 5 minutes, we can finish it. If you have any talk or directly you want to suggest anything, I have unmuted all of you. So now the questions in the chat are over. Kindly, if you have any questions on today's talk, please ask.
My name is Asher. So Jonathan did mention about, Adam and Eve, the possibility of Adam and Eve being the first priestly couple, right? In that case, if we have, let's say, humans outside the garden, and from the emerging population, Adam and Eve is the first couple that is selected, do we have to redefine what we mean by original sin?
No, I would like to answer that. See, there is no need to redefine what's meant by original sin. That's why God clearly said Adam and Eve disobeyed. Obedience is the path to intimacy. When a big person disobeys, that is the one which makes them to move away from God. They moved away from God. That's why God had to curse them. And you can always see the curses existent even now. So God sent them out of the garden. And at that time, whether God sent them alone or sent them along with some more humans, we are not sure. So when we are not sure, I don't want to talk about it. When Bible is silent, let us be silent about it. But I'm sure that we should never redefine that as original sin. Otherwise, Jesus, there is no need for Jesus to come back. Why he should come to this world, leaving he, the God who created everything, Father, Son, and the Holy Spirit, created 2 trillion galaxies, and the whole universe is expanding at a critical expansion rate. With all the things, the God who created everything left and came to this dusty planet to die on the cross. If you believe that, that means you need to believe on the original sin. That's all I would like to say. Anyone else?
Yeah, I read something about the assumptions of evolutions. About there are about 10 assumptions. Can you tell us more about that, if at all? For example, I read somewhere on the internet that says that the first assumption is matter can neither be created or destroyed.
Well, that has nothing to do with evolution, right? So that's the first law of thermodynamics.
Yes. Okay.
I'll post it on the group. Yeah.
So, Prem, you had some 1 or 2 assumptions. That was atheism. Prem was telling about atheism was one of the assumptions behind naturalistic evolution. That he would be addressing next episode. If he can speak on that. That is one of the major assumptions of evolution. Prem, you are muted.
Yeah, sorry about that. So I think that I've been looking at some of the comments, the various comments, and also hearing the person who just asked the question. So what I would like to say is that when we do science, really any branch of science, of course, science has its own principles that we adhere to. Otherwise, there won't be any integrity in the discipline. However, some level of assumptions, some sort of philosophical underpinning, a set of beliefs is always underneath the practice of science. So philosophy cannot be completely avoided in the search for truth, especially when we form concepts. So if someone were to ask, what do you mean by information? Or what do you mean by design? That will require some kind of idea, which by itself doesn't come from biology. So if we said design, the idea of design, that's something that is looked at formally under philosophy. So if we are using that term, then it means that we have some kind of assumption. So for evolution, we think about Darwinism. The whole premise of Darwinism, whether it's the original form held by Charles Darwin, or what we call the neo-Darwinism or the modern synthesis, it's predicated on the belief that there are no supernatural causes. So that is an assumption. To say there are no supernatural causes, or to say that there are only natural causes, is a position in philosophy. It's a philosophical position called naturalism. And so on the assumption of naturalism, if you're going to create an explanation, then it seems you don't have too many options. And as Jonathan pointed out, the theory of evolution seems to be the only option available on the assumption of naturalism. So what I like to do is to try to get people who are thinking about this to consider that there are always a set of assumptions that people hold to, whether you are reading the Bible, or whether you're crossing the road, or whether you're thinking about the origin of life. And so when we understand the assumptions, we can then critique those assumptions and ask, is that a good assumption to have? Is it reasonable to have that assumption? So that falls under the domain of philosophy. And what unites thinking about evolution, or about intelligent design, or about philosophy is the search for truth. So it's not a bad thing, or somehow a deficient thing to say that we ought to think about things like assumptions and philosophical ideas, as if to say that science is somehow superior to philosophy. Actually, it can be shown that science has underpinnings or foundations which can only be properly analyzed under the subject of philosophy. So with that in mind, when we are constructing a critique, some of the critique could be evidential. Some of the critique could be to show that the theory of evolution fails to explain certain features. And then some of the critique could be a more philosophical critique, which is to understand things like the nature of information, or whether naturalism is a good assumption or not. So hopefully that touches on it, which is that, yes, evolution and even science will always have certain assumptions being made. And it's good for us to learn about those as well.
Thank you. Thank you all. I hope there are no more questions. Anybody would like to unmute and just give your comments, you can give. I'm going to switch off in just 2 minutes. Thank you, Jonathan. Thank you, Prem. Thank you so much. It's a wonderful session. It's a wonderful session. So radioactive dating, we will take it up in the next episode. This is a long episode actually. So next time we will try to finish everything in 1.5 hours. So we will try to take it up in the next 2-3 episodes. Let's see. Thank you all. Good night. So good night to all of you. Thank you, Jonathan. Thank you, Prem. Bye.
Good night.
Thank you.
Thank
Good
you.
night.